Hello, this is Abby,
Cindy, and Sam, and we investigated the effect of NHR-23 knockout on C. elegans chemotaxis. C. elegans are free-living nematodes that can be found in soil or collected from decaying vegetable matter. Researchers commonly use them as model organisms in the study of animal behavior. This is because they have a simple nervous system that has been entirely mapped out by researchers and possess the ability to sense a wide range of environmental stimuli. C. elegans chemotaxis assays are commonly used to investigate nervous system activity. Chemotaxis is the process by which an organism senses an attractant molecule and responds by migrating directionally toward it. C. elegans accomplish this through the use of 32 different chemosensory neurons.
While a fully functioning nervous system is very crucial to the development of C. elegans, the molting process is equally as important. Molting is the process by which the old exoskeleton, also referred to as the cuticle, is replaced by a new one. Studying this process can provide valuable insight to extracellular matrix remodeling, intracellular trafficking, and even signal transduction pathways. The cuticle also attaches to muscle cells within C. elegans, allowing for facilitation of movement. The molting process is essential for the development of many animal species, and failure to molt properly can result in physical defects.
Molting in C. elegans is regulated by a range of environmental and genetic factors, one example being NHR-23. NHR-23 is a nuclear hormone receptor that is known to regulate several types of collagens and hedgehog-related proteins, which are proteins that are required to be fully functional in order to ensure normal molting. Previous studies have shown that high levels of NHR-23 mRNA are present during intermolts in C. elegans, suggesting that the receptor is activated at the initiation of the molt, ultimately stimulating a molting cascade. Because NHR-23 is crucial to the beginning of the molt process, its loss could have significant effects on the physical appearance of the worms. The absence of NHR-23 results in a worm that is shorter in length compared to wild-type worms, which may be due to the misregulation of a specific collagen gene.
For this study, we wanted to investigate whether or not the loss of NHR-23 has any effect on C. elegans chemotaxis. Since we know that NHR-23 absence results in worms that are shorter in length, we have reason to believe that use of NHR-23 RNAi would yield this exact result. As mentioned in a previous slide, study of the molting process in C. elegans can provide valuable information regarding signal transduction pathways. We believe that the change in the molting process brought on by the knockout of NHR-23 will have an effect on signal transduction, ultimately compromising the process of chemotaxis. We also mentioned in another slide that the cuticle attaches to muscle cells within the worms, allowing for facilitation of movement. Improper molting may disrupt these connections, causing the worms to move less efficiently. Because of this, we think that chemotaxis in the mutant C. elegans will be significantly reduced or inhibited. Therefore, investigating the effect of the knockout of NHR-23 on C. elegans chemotaxis will not only provide confirmation that NHR-23 plays an important role in molting, but will also demonstrate that molting defects in an organism can compromise otherwise normally functioning processes. As far as we know, the nervous systems of the worms used in the experiment are fully functional, so any significant differences in chemotaxis between the experimental groups would be due to improper molting. This could open up new doors to further research on the effect of improper molting and other physical defects on C. elegans, signal transduction, and other processes.
NGM plates were made using these ingredients, and RNAi plates were made using the same ingredients, also with ampicillin. The plates were left to sit for a few days to make sure that there were no contaminations.
Next, bacteria were grown so that the worms could have a food source once placed on the plates. The control plates had OP50 E. coli, and the RNAi plates had NHR-23 knockout bacteria.
8 to 10 adult worms were placed on each of the 4 bacteria plates and left for a few days to produce offspring that would have a phenotypic change in response to the NHR-23 knockout. Worms were washed and placed onto the chemotaxis plates using the C. elegans protocol that we received in class.
The setup for the chemotaxis was also the same as the lab protocol that we had previously done. Once the C. elegans had an hour to move, they were counted. The length of the worms was taken using the dissecting scope after chemotaxis was completed.
The Chemotaxis index was calculated, and the length of 32 worms was recorded and compared by doing a t-test and SPSS. A univariate ANOVA was also done using SPSS to determine if there was a significant difference in the chemotactic ability between the NHR-23 and control worms.
To determine if the knockout was successful, the C. elegans were examined under the dissecting scope in Dr. Cooper's lab. Pictures were taken to compare the body length of the two groups. On the left in Figure 2 are the normal C. elegans, and on the right in Figure 3 are the NHR-23 knockout C. elegans. Under the dissecting scope, there did not appear to be any difference in the worm's body length.
The length of 32 worms was recorded and the mean, plus or minus the standard error of the mean, was calculated to determine if the NHR-23 knockout was effective in altering the phenotype of the C. elegans. The results of the t-test reported that there was no significant difference between the C. elegans mutant length and the control length because the p equals 0.272, which is seen in Figure 4. These results suggest that the knockout was not successful because it was expected that the mutant C. elegans would have been shorter than normal.
To test the effects of NHR-23 knockout on C. elegans chemotaxis, a chemotaxis assay was done using the mutant C. elegans that had the knockout and a control C. elegans that did not. Diacetyl was used as the chemoattractant for each. The results of the ANOVA using the chemotaxis indices reported that there was no significant difference between the control and the mutant C. elegans, with p equaling 0.333. There was not a significant difference in the movement of the C. elegans toward the diacetyl. These results also suggest that the knockout may not have worked because there was no difference in the chemotactic ability between the two groups.
Based on our statistical analysis, we determined that there was no significant difference in both body length and chemotaxis index between the two experimental groups. We originally predicted that a successful NHR-23 knockout would result in C. elegans that are significantly shorter in length. This hypothesis was not supported, as we believe the knockout was unsuccessful. If the knockout had worked, we would have expected to see results similar to another study that showed that reduced levels of NHR-23 resulted in significant molting defects. We also hypothesized that chemotaxis of mutant worms would be significantly inhibited. Our reasoning for this was because of the fact that the C. elegans cuticle is attached to muscle cells within the worms, allowing for easy facilitation of movement. We thought that any irregularities in the molting process may disrupt these muscle cell connections, reducing efficiency of movement and eventually inhibiting the process of chemotaxis.
If we were to replicate this experiment again, the first improvement is to put more C. elegans on the NHR-23 knockout plate initially to increase the likelihood that they will produce enough offspring that have the mutation. We would also measure the worms right after the chemotaxis is done or before chemotaxis begins to see if there is a difference in length, and finally, we would watch the C. elegans before chemotaxis to observe if there are any differences in movement.
For future research, the experiment should be replicated, but allow for more time for the original worms on the RNAi plates to feed and produce offspring to increase the likelihood of having offspring that have the NHR-23 knockout. This adjustment in time, along with changing the length of time for the chemotaxis itself, could help determine if the amount of time given to move plays a role in the worm's ability to move. Other future research should be done to knock out other nuclear hormone receptors that are known to affect C. elegans molting to determine if molting does in fact affect C. elegans movement.
So now the only question that remains is, what is the broader significance of all of this research? Well, if we were to replicate this study in the future and achieve the expected results, that is, if chemotaxis is inhibited by the absence of NHR-23 and C. elegans, then this could provide more opportunities to study C. elegans in the future. Researchers know quite a bit about C. elegans' nervous system functioning and how the different neurons mediate different chemotactic responses. In this case, Scientists would have the opportunity to investigate how physical defects rather than nervous system defects can affect signal transduction pathways and any other processes that we know are otherwise uncompromised and fully functional. Overall, researchers could have more resources in the usage of C. elegans as a model organism.